Hi, and welcome to this update by Oxford Nanopore Technologies. My name is Rosemary, and I'm calling live from ASHG 2025, all the way from Boston. I'm here to give you a little update about how Oxford Nanopore is seeing the space of human genetics today. Our ability to read the genome has evolved very, very significantly in the last 70 or so years, all the way from early gel through microarrays, short read sequencing, and now the era of long, fully loaded genomes. Despite this amazing development, clinical pathways are still really long and very, very cumbersome, with many families waiting years to get an answer and us leaving an enormous amount of information still on the table. And when we look at why this takes so long and why this is so difficult, it's not a surprise. Genetic testing is multifaceted. So many different types of variants contributing to complex disease, be they SNPs or indels, copy number variation, structural variants, methylation, imprinting disorders. All of these contribute hugely to the diagnostic yield. And despite all of the amazing technology that we have developed together with the community over the last century, all of these technologies still leave gaps. And so you still have a lot to do in terms of getting to that high diagnostic yield. So let me talk to you a little bit about the Oxford Nanopore platform. We have created a completely novel method of sensing DNA, and we hope that with this platform, we can help address some of the challenges that I've just spoken about. At the heart of our sensor is a nanopore, and what we do is we take DNA or RNA and we stream that molecule through the nanopore with our motor enzymes. We work with the latest and greatest in machine learning and AI to translate the raw signal into bases, and we can scale this core tenet of our technology onto smaller flow cells like our MinION flow cells or larger flow cells like our PromethION flow cells. And we can scale this yet again onto our instruments through distributed instruments, benchtop instruments, and high throughput instruments. Reading molecules in this way, so doing this direct sequencing of DNA or RNA, means that we have richer information, including epigenetics. We can read any read length, any analyte, which means that you only need one platform for any sample type. You can get real-time analysis for rapid turnaround times. And because our systems are so modular, you can sequence on demand without a batch penalty. This method delivers data in a completely new dimension. The richness of information that you're now getting from nanopore sequencing is enabling people to map the areas of the genomes, call structural variants, call SNVs. We have basic DNA methylation like 5MC and CpG contexts, but we can go way beyond that with advanced DNA methylation, RNA methylation. We can then phase your variants and also assemble genomes. So with all of this technology, we have many customers driving novel insights and novel discoveries. And I'm going to go through how this is happening in the human space. Our human product range really centers around PromethION. And we have this in two different options. We have a PromethION 2 and we have a PromethION 24. And this is real scalability of our platform here. So typical outputs now for customers, typically just above about 30x for one flow cell. And so on a P2, you're talking about 200 genomes per year. On a P24, we're scaling this further, and you get to about 2,500 genomes per year when you're running one human genome on one flow cell. We have developed a really rich number of different human end-to-end workflows that deliver complete soup to nuts, so extraction all the way to analysis. And we have methods for targeted sequencing, low pass, standard 30x coverage, and also T2T. And we start from many different sample types, all the way from blood, swab, saliva, and many others. And you can find all of this information on our website. We've been working on pushing scale for human genomes for many years, starting with our Emirati genome program, where we've now done over 100,000 genomes, and of course, working with groups here in the States like the 1,000 Genome Project, and around the world like Precise, NIHR, Genomics England. And we've really worked with these collaborators to really fine tune our platform and get all of the best information that you can out of the system. Our workflow for high-end large cohorts has two core components to it. We first of all create a reference assembly for the study using T2T, and we then scale that with a 30x workflow. And if you look at our T2T assemblies, Nanopore now enables a single platform, telomere to telomere assembly. All you need to do is run our ultra-long data. You can then phase that with some PORC data and run the latest tool chains that have been made available by the community. Using this methodology, you can get 46 out of 46 T2T scaffolds all on one platform. And if you look at how this performs versus the typical hybrid approaches, you can see that the nanopore-only method is delivering superior performance across the standard. So now that we've created a backbone to the study through the T2T, we then scale that with 30x high throughput sequencing workflows. We've been doing this now for a number of years, so we have a very, very well-developed sample prep methodology, which includes automation. We have our sequencing methodology with our PromethIONs and our PromethION flow cells. And we have a very, very developed analysis pipeline, which goes from your base calling to your BAM files through epitome human variation to get to a tertiary analysis provider. In the example here, we can see one of the studies, one of the customers that we've had, typically generating 130 to 140 gigabases on average, and with over 90% of the runs delivering over 100 gigabases. But we really believe that we can do better than this. And we have a very strong R&D roadmap to deliver lower costs per 30x genome. And not only will we're looking to solve two things. The first thing is getting this genome more accessible. But the second thing is making our platforms, the P2s and the P24s, more highly scaled. So every time we increase the output of our flow cells, the scalability and the number of genomes that you can run per instrument also increases. And I'm going to tell you about three key things that we're going to be doing in the next year to deliver some of these improvements. So our first enabler is the ability to load multiple genomes on single flow cells so that you can pull multiplex across an instrument. And for that, we have a new kit in development called the high throughput barcoding kit. So you barcode your libraries, pull them, and you can run 24 samples across 24 flow cells. And what you can see here is internal data that shows that when you start multiplexing and pooling them, you get over 99% of your genomes now hitting 30x. So it removes any variability that you may have in output. But then you can also start putting 24 samples, for example, on 20 flow cells or 16 flow cells. And that starts bringing your price per genome down. We can see that we have this kit now in place with beta testers, and you can see here the first results coming out, 347 samples processed, all of them getting to over 90 gigs. And this is in beta testing now for release in 2026. So that's your first enabler of how to get a lower cost per genome. Now let's talk about the second enabler. We have to increase the output of our flow cells. And now this is something we've been working on for a very, very long time. One of the core areas where we suffer from output is pore blocking. Many of our customers are familiar with this. And our teams have really worked so hard to deliver an improvement in our flow cell that reduces the amount of blocking that happens. The beauty of this is that it's the same nanopore, it's the same base calling models. All you're doing is getting a new premium flow cell, the PromethION Plus, and loading the current libraries that you load today. You're going to see a 30% enhancement in output when you're running a 15 kb. It will also reduce the number of interactions, washing interactions that users need to do on a regular basis. These flow cells are out with beta testers now. We're going to be rolling into an early access during Q4 and scaling this up in 2026. So that's our second enabler. The third enabler is higher output chemistries. So now that we've solved this blocking issue that we've had, we can start exploring all of the latest innovations that our R&D teams do around enzyme design, machine learning enzyme design, to get us a higher output chemistry, so generating more data per run. And we can see here that when we add the improved flow cell chemistry and we add a higher output chemistry on top, we are now talking to close to 70% increase in output. So pushing the envelope towards those 200 gigabases per flow cell. Again, we've got a beta during Q4 and a scaled rollout through 2026. With this product, we are initially targeting high throughput human applications. So hopefully you can see here that with these three enablers that will be moving very, very swiftly during the next 12 to 18 months towards a 200 gigabase flow cell, which then gets you a 30x genome for about 345 for those high throughput applications. But don't worry, we are not stopping there. Our R&D teams continue to work on every single area and every single nugget of our platform, be it raw read accuracy, consensus accuracies with future nanopores, and even more output. So the message is always, if you invest in Nanopore, we invest in you, and we will continue to deliver amazing product upgrades as you work and collaborate with us. So I've talked about how all of our higher throughput customers are generating all of these novel insights, all of these novel discoveries. But how about the application of them? How about the go? How are we really translating all of these novel insights into clinical action? There are now over 50 studies that show incredible evidence that utilizing long reads or utilizing Nanopore can significantly boost your diagnostic yields. All of these have been done in either independently or in collaboration, and they are phenomenal pieces of work. Again, we're using the same workflows that we've developed for our research, for our high throughput end. But really, when you start getting into the clinic and into the LDT space, targeted sequencing begins to have a real power. And at Nanopore, we have a very, very unique way of performing targeted sequencing that I want to tell you a little bit about. So it's something called adaptive sampling. Many of you know of targeted sequencing in PCR panels where you're doing 10, 20 genes, or hybrid capture panels where you may be doing 50 and 100. Adaptive sampling panels are the most comprehensive targeted sequencing panels. They are completely sample prep free. You don't need to do any enrichment up front, and you can capture hundreds of genes. So it's incredibly comprehensive. So let me tell you a little bit about how this works. What you do is you create a reference file with regions of interest in the genome, and you upload that into our software, into our Minnow software. As the DNA approaches the pore and starts to get sequenced, it will automatically, in real time, align against your file. And if it's a region of non-interest, it will get ejected by the pore. If it's a region of interest, it will continue to get sequenced. And what this results in is enrichment of these areas of interest. So you might be getting 30, 40, 50, 60x coverage of your regions of interest. But we're also still sequencing all of the parts that are getting ejected. And so what you're creating is a 1 to 2x whole genome backbone underneath this. And even in that low coverage, you can detect chromosomal gains, losses, polygenic risk scores, and many other types of variants. So you're really getting a complete picture. And remember, there's no sample prep to this. You are doing a whole genome library preparation. You are loading that library preparation in, and you are then sequencing. So let's talk about some of the panels that we have. We've been packaging these up into pre-made workflows. The first one, tumor methylation profiling using adaptive sampling, that is 723 genes covered, full variant detection, and customers are getting methylation classification in 1 hour. And this enables intraoperative brain surgeries to be highly guided by nanopore sequencing and having huge patient impact. You can carry on the run, you can let it carry on so that you get a pan-cancer panel in 72 hours. On hereditary cancer, we've just launched a hereditary cancer panel, 258 genes covered, again, full variant detections, SNVs, SVs, copy numbers, methylation, full long read, enabling your variant phasing. And here you can load 3 genomes per flow cell. And you're getting a sample to answer in about 96 hours. Again, no upfront lab work to do. And then pharmacogenomics, 375 genes covered, full variant detection, 4 samples per flow cell, 72 hours. And here we're covering really challenging genes, including things like CYP2D6. A really important part of adaptive sampling is if you decide that you want to adapt your panel, you want to add a gene, all you do is program that into software and then run your sample. There's no need for an enormous amount of lab optimization. It is the most dynamic panel that you'll find today. So all you need to do is switch it on in software and press go. And I hope you have fun with it. Now, what this does to your sequencing capacity is what you're now doing is 4 samples, for example, 4 targeted genomes on 1 flow cell. So your P2 all of a sudden is going through 800 targeted genomes per year, and your P24 is now at almost 10,000 targeted genomes per year. So again, the scalability of our instrumentation just continues to shine. There are some times when you need the full genome. You don't have a closed question, you need open, particularly in critical care settings in stages like NICU, PICU. And for this, we've just launched our 24-hour full genome workflow. It's the whole genome, full variant detection. We're using 3 flow cells per genome to get the time element there, and your sample to answer is 24 hours. So I hope you can see that through all of the different work that we've been doing and through all the different studies that all of our amazing customers and collaborators have been doing, we can demonstrate now that Oxford Nanopore can start to replace a multitude of different technologies and get those patients the answers that they so desperately need very quickly. So our platform is one of boundless opportunity. We can sequence it and we can scale it to any scale. We can put down any biology. I know I've spent the last 10 minutes or so talking about DNA on direct RNA. There are amazing breakthroughs coming through around the impact of RNA modification and how this plays a critical role in disease. And so please do look up many publications coming out here to get inspired. And also on the proteomic side, our amazing R&D teams continue to accelerate our path towards having a protein sequencing product in the not too distant future. So with that, I'd like to thank you for listening and thank you for all of our amazing customers that deliver all of these amazing insights and help us really get the complete picture. So thank you so much, and I look forward to seeing you soon.